We previously discussed characteristics of living and non-living things, and your students should be able to differentiate between the two. They should also realize the importance of classifying and ordering both living and non-living objects according to their similarities and differences. It not only gives everything an orderly arrangement, but it allows us to study and compare objects within each group. There are many activities that you can use to help introduce this concept of classification. One is to make an assignment where your students go out and look for ways in which we classify objects. They may come back with ideas like library books, silverware in a drawer, clothes in a closet, or food in a refrigerator. And then you might even ask them how we classify those particular objects. In other words, what characteristics do we classify them by? Library books, for example, we don't classify by the size of the book, but rather by the subject matter of the book. Another good activity is called Name the Mystery Student. This time you do it in class. Pick some characteristics of one particular student before you start the activity, and then write one characteristic at a time on the blackboard. For example, your first characteristic might be the student is a boy. Who is the student? Well, you've only narrowed it down to about half the class. Characteristic number 2 may be the student is wearing tennis shoes. Well, we're starting to get it narrowed down to fewer and fewer students in the classroom. Eventually, with more and more bits of information, the rest of the class can identify who the mystery student is. The key to this is to start with general characteristics first and then narrow it down to more specific characteristics. There's some other good activities that you can do with worksheets. The first, each student circles 2 objects out of 3 that are most closely related. You might throw in some tricky ones like shark, horse, and gorilla, or tree, mushroom, and rose bush. And don't forget non-living items too, like pencil, pen, and eraser, or motorcycle, car, and bicycle. In the second handout, we will again have the students classify different objects, but this time the shape of different objects that are on this piece of paper here. This time, have your students cut out all 13 objects and then classify them according to their shape. Our first general classification we will call kingdoms. We'll have 2 kingdoms. The first kingdom will have object number 3, 4, 6, 7, 9, 10, and 11. That will be kingdom A. Kingdom B will be the remaining ones. Let's now ask the students what are the characteristics that separate Kingdom A from Kingdom B. And also, what name might we give all the objects in Kingdom A? Well, I would say a good word might be notched. And for Kingdom B would be no notch. Then you want to take all of the objects in Kingdom A only and classify them separately into subgroups which we'll call phylum. The first phylum will be items 3, 4, 7, and 10. The next phylum will be items 6 and 9. And the third phylum will be item 11 all by itself. Once again, have your students identify characteristics of each group and give each group a name. In this case, we might call the first group round notch, the second group square notch, and the third group diamond notch. Then we want to take the first phylum and break it down into subgroups. This time we'll call these groups classes. In this case, we have 2 classes. The first class is items 3, 4, and 7, and the second class is item 10. Once again, the characteristics and the name. In this case, it could be round single notch and round double notch. If you wish, you can repeat the above activity with Kingdom B and have your students go through the same exercise. Here they're learning about classifying different objects according to their characteristics. Your students should also be aware of the great variety and diversity of living things and realize the necessity to classify these life forms in order to study them. You may wish to next introduce the classification scheme that we use in biology. The levels are kingdom, phyla, class, order, family, genus, and species. A good aid to remember this is King Paul cried out for good soup. Using this order, every living thing will have one scientific name known the world over. It's like an address. We live in a country, in a state, in a city, on a particular street, at a particular number, and we have a surname and a first name. All of this information identifies who we are. In life science, we use Latin words to identify different life forms. These are the genus and the species names. The genus is capitalized and it's a noun. The species is a word that follows it, and that's usually an adjective. For example, if we look at cats and dogs, they both fit into the same kingdom, phylum, class, and order, but they fall into different families, genus, and species. If we identify an animal as a Felis domesticus, we're talking about a common house cat. A Felis tigris is a tiger, and a Felis leo is a lion. Incidentally, a Homo sapien is a human being, and the word sapien means wise. At the top of the classification scheme are the kingdoms. Some scientists see only 2 kingdoms, plants and animals. Others see 3, plants, animals, and protists. Still other scientists see 5 kingdoms, plants, animals, fungi, the simple single-celled organisms like bacteria, and then the more complex single-celled organisms like the protozoans or the paramecium that we previously looked at. Textbooks are still split on this 3 or 5 kingdom classification, but the important factor, the important point, is that there are many life forms that don't quite fit into the plant or the animal category, so they need to fit into their own separate little niche. These life forms include the protozoans, slime molds, fungi like mushrooms, mildew, and yeast. Also there's lichens, blue-green algae, bacteria, and molds. Most of these life forms fall into this category because they're unable to make their own food, like the fungi. The slime mold is a very interesting example in that one stage of its life, it's a mass of cell material with no cell wall, but many nuclei, and it actually has the capability of moving along a log with an amoeba-like motion. Let's now investigate some of these simple life forms with some interesting activities. A fungus is a plant-like organism that survives in one of 2 ways. It either lives off another living organism as a parasite, or it survives by living off dead organic material. Like a plant, a fungus has cell walls and cannot move about. But unlike a plant, a fungus does not have chlorophyll. It cannot make its own food. We will start by investigating the mushroom family. Unlike other types of fungi, mushrooms have their spores growing in a club-shaped structure, so they're sometimes called club fungi. There are many different types of mushrooms, and you should caution your students about eating mushrooms they find growing in the wild. Only eat the store-bought type. One type of mushroom you might find growing on a dead log or a dead tree is called the bracket fungi, and it grows on the side of the tree like this. And you can peel them off, break them off, bring them to class to show to your students. Another type of fungi is the common type that we find in the grocery store. You might bring a dozen or so of these into class and have your students take them apart and look closely at the different parts of the mushroom. It's really quite interesting. There are 3 main parts. The top part is called the cap. Inside the cap, where all the spores are located, is called the gills. And then the stalk that comes out is called the stipe. Your students can use their magnifiers in this activity, make good observations, and record their results. You can also have them split the stipe in half and look at the interior structure. The next activity is using mushrooms, and this time we want a mushroom that's fairly old, where the gills are exposed. In other words, the cap is opened up. You may even want to peel back little pieces of cap that may be covering up the gill structure and snap off the stipe so that you have a mushroom that looks something like this. Take the mushroom and place it face down on a piece of light paper and cover it with a plastic drinking cup. This is to keep the air currents from blowing the spores around as they drop off of the gills. Let it sit like this for 24 hours. After 24 hours, pick up the cup and remove the mushroom cap. You'll find a very beautiful spore print of the mushroom that looks like this. This is a very delicate and fragile item and should be handled carefully. Your students should realize that there are literally thousands of spores on this one piece of paper, each capable of producing a new mushroom. If you want to preserve this spore print, you can spray it with some art fixative, or you might even try hairspray. Now let's look at the next type of fungi, the molds. These are fungi that have their spores in special spore cases. Characteristics of this mold type of fungi are long thread-like structures with a little spore case at the top. When the spore case bursts open, it allows the mold to spread quite rapidly along the food surface. Remember that molds are not true plants because they can't make their own food. We're all familiar with molds on certain foods, and who would believe we'd be growing them on purpose in the classroom? But this is what we see on some cheese. We'll now do an experiment to find the best ideal conditions for growing mold. We'll start with two pieces of bread, one with no preservatives and one with preservatives. We'll cut each into 4 pieces, and we'll put 10 drops of water on each one of the pieces. We'll then put each slice in a plastic bag and subject each to different environmental conditions. Our first 2 samples, the preserved and unpreserved bread, will go in a warm, dark, open place. In other words, the bag is open and it's in a warm, dark place. Our next 2 samples will be in a warm, dark place with the bag closed. Our third set of samples will be in sunlight with the bag closed. And our fourth set of samples over here are what we call our control. These are in the refrigerator with the bag closed, so it's cold and dark. You may wish to continue and experiment on other food sources too, like coffee grounds or fruits and vegetables. I have a grapefruit rind in this plastic bag. Now let's go on and look at the next type of fungi and the final type. This includes the yeasts and the mildews. These fungi have their spores in tiny little sacs. We've all seen mildew before. It lives off cotton and leather and sometimes the walls of our house. Here you see it on this boot. It's actually eating the leather of the boot. And if you've ever left clothes in a dryer and the clothes didn't get completely dry, you may have noticed the appearance of mildew. It's important to understand the conditions that help mildew to grow so we can prevent them. This next activity will show your students just what conditions are necessary to grow mildew. We're going to have them take 2 socks. The first sock they'll wear, let it get dirty, wear it for a day, sprinkle a little water on it, wring it out, and put it in a plastic bag. They'll store it in a warm, dark place. Sock number 2 will not be worn, but you will sprinkle a little water on it, wring it out, put it in a similar plastic bag, and store it also in a warm, dark place. Have your students observe their 2 socks each day for about a week and make observations. Look for the formation of mildew. Let's now see how our mold and mildew experiments look sometime later. It's been 12 days now since we started our mold and mildew experiments, and boy do we have some interesting results. First, the mildewed sock. The dirty one has more discoloration than the clean one. You might have your students open the bag and smell each sample to detect the odor of mildew. The cheese has gotten much worse in its moldy appearance, and the grapefruit rind has shown a real appreciable change as mold is grown extensively. Now let's evaluate our mold samples. Remember the preserved and unpreserved bread. In all 4 cases, we see that the samples with the preservatives show no signs of mold. But look at these samples with no preservatives. First, the bag that was open in the warm, dark place. It has a green mold growing on it. And the other sample in the warm, dark place, where the bag was closed, has both a black mold and a green mold. The sample that was in the sunlight shows very little growth of mold, and the sample in the refrigerator, our control, shows no growth at all. There are a number of ways you can do this experiment. One is a demonstration in the classroom, and after 12 days or so, your students can go around and make the comparisons. Another way is to have your students do this experiment at home. Make the samples in class, take them home, store them for 7 to 12 days, make daily observations, and then after 12 days bring them all back to class, group them all together, and then have all the students walk around and look at samples of each group. They should conclude from doing this experiment that there's a best place to store bread to keep it from molding. They should also learn about the action of preservatives in keeping mold from growing on bread. One of the best ways to learn about yeast is to work with it, experiment with it. And this experiment is designed with you in mind. We're using lots of simple materials. It's good for learning about yeast. There's a lot of different areas we can branch out to, and the students are going to be learning scientific methodology at the same time. Here's what we're going to try and find out. How does yeast react when it's in a sugar solution? And does the concentration of the sugar in the solution affect the amount of reaction the yeast makes? We're going to be using these small soda bottles, and you should have one for each group of 3 students. The other thing you'll need is a device for transferring any gas produced in this bottle over to another container. There's 2 ways to do this. One is the scientific elaborate glassware device, and this is made very simply by heating up a glass tubing in the fire like we did earlier, and this time bending it to a 90-degree angle and letting it cool. After you've done this corner, you go on and do this corner, and you make a little gadget like this. When it's placed in this jar, and there's another container over here that the tube sets in, any gas that's produced in here will go across and bubble through in the second container. If you don't have the capability of making one of these, you can make a simpler device out of a piece of aquarium tubing. This is from an air pump, and a piece of modeling clay. Roll the clay out so it's very thin and long, and then carefully wrap it, kind of like a cinnamon roll, around the aquarium tubing, like you see here. Pinch it down so it makes a real tight seal around the tubing, and then jam it into the top of the bottle. You want it, once again, a very tight seal here, otherwise gas will seep out around the edges. Now we're ready to mix the sugar-water solutions. We'll start with 100 milliliters of warm water. Use the graduated cylinders and let your students get experience making this volumetric measurement. If you don't have these available, you can substitute a half of a cup. Once the water's in each jar, you want to add the proper amounts of sugar. Make sure that each group has a different concentration of sugar. The best way to make the sugar measurement is to weigh it. Get a piece of filter paper and weigh the filter paper all by itself. This piece of filter paper weighs 1/2 gram. And then move the balance up to 5.5 grams and put sugar on the paper until it comes into balance. You know now that you have 5 grams of sugar sitting on that piece of paper. Very carefully transfer it over to the right bottle, put a little fold into it, and shake it into the bottle. Make sure that one group has 5 grams, another could have 10, a third group could have 15. You also want to add yeast to each container, and a good amount of yeast to add is 2 grams of yeast per bottle. You can use the same type of procedure to measure out the yeast, and each packet of yeast has about 7 grams in it, so this is good for 3 groups. You also see these 2 containers on the end are not sugar water at all, but one of them is apple juice and the other one is grape juice. We already know that these fruit juices have sugar in them, so we don't need to add sugar to these 2 containers, but we will add the 2 grams of yeast to each sample. Now, if you don't have the balance available, you can substitute teaspoon measurements instead of weight measurements. The yeast, 2 grams of yeast, is about 1/2 teaspoon, and 5 grams of sugar is about a teaspoon, so you can always do it that way too. Now, nothing's going to happen right away. So after your students mix their solutions, you should put them in a warm place in the classroom and let them sit for about an hour. After the hour is passed, we'll do our science experiment. With the magic of video, the hour is quickly passed and we're ready to continue our experiment. Your student group should first compare one sample to the next and notice that some are frothing more than others. There appears to be more bubbles in one than another. Look at our control. This one could be your sample. It didn't have any sugar in it, but it had the water and the yeast, and there doesn't seem to be any reaction at all. It appears that the yeast needs sugar to react. If we had just sugar and water together, we wouldn't get any bubbles. So it appears that the yeast is reacting with the sugar and the water. Now what is really going on here is a process called fermentation. It's the means by which these simple organisms break down sugar and use it for energy and make alcohol and carbon dioxide as a byproduct. So there's a chemical change taking place here. It's a biochemical change, and we can tell that this chemical change is taking place because gas is being produced. The next question we might want to ask is, does the concentration of sugar affect the amount of gas that's being produced? In other words, does a group with 10 grams of sugar in their bottle get more gas produced than one with 5 grams? Well, the way we'll do that is with our little containers of liquid and our little transfer tubes. We'll set this one here has 5 grams of sugar, and we'll set this one next to it with 10 grams of sugar, and we'll compare the bubble rates. Another thing you could do is have each group count the number of bubbles in a 1-minute time period. They could do this over a 10-minute length of time. Write down the number of bubbles in each 1-minute interval, and then we can compare the values from one sample to the next and find out if in fact the more sugar in the container means that there's more gas produced. And then we can go on from here even and make a comparison as to whether this grape juice is equal to a 5-gram or a 10-gram bottle of sugar. In other words, we're finding the relative amount of sugar in the grape juice or the apple juice by comparing the bubble rate. So you can see there's really a lot of ways you can go with this one. Let's take a close look at these bubbles and see how fast they're coming out. The bubbles from the tube on the right appear to be coming out at a faster rate than those from the tube on the left. This would suggest that the greater the concentration of sugar, the more gas that's produced. To make this experiment more exciting, instead of using just colored water here, we'll use a chemical called bromothymol blue. It's an acid-base indicator, and when mixed with tap water, it turns the tap water blue. This means that your tap water is a little bit on the basic side. If it happens to turn it yellow, throw in a pinch of baking soda to make it turn blue. Then we want to bubble the carbon dioxide through the bromothymol blue water. After a while, the carbon dioxide combines with the water and forms a mild acid and changes the color of this water from blue to green and then to yellow. Another thing you can do is use this special test paper. It's designed to test the presence of sugar, but only simple sugars like glucose. It won't work on cane sugar, sucrose, the common type of sugar we're used to. So if we tested this piece of paper on regular sugar solution without yeast in it, we would see that it wouldn't change color. It'll stay yellow. But if we test it again on a solution that the yeast has already acted upon, we'll find that there's simple sugars in this container. And by doing this, we'll find out that the yeast somehow changes the chemical structure of the sugar. It breaks down a complex sugar and turns it into a simple sugar. And as you see here, we're getting a reaction. We're seeing presence of simple sugars in this solution that used to be only complex sugars. So the yeast is somehow changing the form of the sugar. I hope you can see all the benefits in doing this experiment. We cover measuring, process skills, an understanding of fermentation, how yeast breaks down complex sugars to simple sugars and then to alcohol and releases carbon dioxide. If you want to look at the yeast under the microprojector at the beginning and then a day later, you'll see many more yeast cells. They're reproducing by the hundreds.